Right, welcome everybody. This is the third in our Meet the Pro workshop series. And today I'd like to welcome Dr. Madelaine Bartlett, a plant biologist at the University of Massachusetts. Dr. Bartlett is Associate Professor in the Department of Biology at University of Massachusetts Amherst, and she is also the Director of the UMass Natural History Collections and the Bartlett Biology Lab. She earned her bachelor's in molecular and cell biology, as well as a bachelor's with honors in genetics and developmental biology from the University of Witwatersrand, South Africa. She earned her PhD in plant biology at the University of California, Berkeley, and did her postdoc in biology at Brigham Young. She is the recipient of the 2019 UMass Lilly Teaching Fellowship and was nominated in 2018 for the UMass Distinguished Teaching Award. In 2018, she received the Botanical Society of America Emerging Leader Award, and in 2016 was a nominee for the Packard Fellowship Competition. In 2015, she was awarded a UMass Student-Centered Teaching and Learning Fellowship. As director of the Bartlett Lab at UMass, she and her team study the evolution of plant development. They work towards uncovering the molecular mechanisms that control the development of plant form, and then understanding how these molecular pathways have changed over the course of evolution to generate morphological diversity. One aspect of their work is studying the genes and gene networks that have important impacts on crop productivity. Welcome to our meeting today, Dr. Bartlett.
Hello, everybody. Thanks. This is fun to be here. And I'm going to tell you a little bit about myself and what my journey was to get here. So I'm going to start sharing my screen.
Go right ahead.
Can you see my screen?
Should be coming up. There you go.
Okay.
Perfect.
So this, I was thinking about what to call this, and I think this is about, the title is really about what encapsulates why I ended up doing what I was doing, right? And so I'm going to talk a little bit about how I got to do what I'm doing now. So as Matt said, I'm from South Africa. I was born in Johannesburg, South Africa. And this is me when I was a kid. This was my favorite activity.
Love it.
You'll notice I'm not athletically swimming across the pool or anything like that. I'm just sitting on my lilo and hanging out in the sun. And this basically is what I did, right? I was not a very good student. I always did well in school, but I wasn't very studious, if that makes sense. I would do the bare minimum to get ahead and spent as much time as I could swimming in the pool and reading books. I like to read. Those are the two things that I did, and were the two things that I was good at. And when I graduated high school, the way that it worked in South Africa then is that, and it's still pretty similar to this now, is that all of our universities are the same. It's not like it is here where there are state schools and there's a lot of choice in which universities you go to here. That didn't really happen in South Africa back then. And really it was just you went to the university that was in your hometown unless you were rich, in which case your parents could afford to send you somewhere else. And my parents were not rich, so that meant I had to go to the university in my hometown. And I had to choose what a major was. And I was either going to do a Bachelor of Science or a Bachelor of Arts. And the Bachelor of Arts is what I really wanted to do because, as I said, what I really liked to do was read books. And so I thought an English degree meant that what I could do was just read books for 4 years and get a degree. But my dad informed me that if that's what I wanted to do, he wasn't going to pay for university. So I instead signed up to do a Bachelor of Science. And that was the amount of thought that went into it. I wasn't a very good student, as I said, right? I was not a model student by any stretch of the imagination, but I got into university and I discovered that I actually really liked science. Oh, and I actually really liked science. So I did as Matt said, I did my degree at the University of the Witwatersrand in Johannesburg. It's in downtown Johannesburg. And there I did a BSc in molecular and cellular biology, and then an honors thesis on the population genetics of HIV. And this was, I really liked this. It was something that I loved learning cool things about how plants work, about how organisms worked, and particularly about the molecules that control how biology works. But when I came to the end of my degree, I was really kind of at a loose end. I was still staying at home in my parents' house with my mom and dad, and I didn't want to live at home anymore. That was, again, what was kind of normal back then, is that people stayed at home during their degrees, and I was sick of it. Mm-hmm. And I also been at the same university for 4 years and in the same town that I'd grown up in for the last 22 years, and I was sick of it, and I didn't know what to do. And so my plan was to go to Europe, to England. I have had some very good friends that were living in England, and my plan was to go and sleep on their couches and be a waitress. And then, and the plan was to work illegally, pretty much, in England, bumming and make enough money so that I could go and bum around Europe and then go back to England, and when I ran out of money, and earn some more money again after that point. And I said this to my honors thesis advisor, and she was like, well, that's one idea. Or another idea is that you could, you seem to really like, my honors thesis advisor taught a plant development class when I was an undergrad. And it was the reason why I chose her as my thesis advisor, because I really, really loved the plant development class that she'd given me. But at that stage in her career, she'd shifted over to population genetics of HIV. And so that's, and she was like, well, you really like that plant development class. There's a lot of really good plant developmental biologists at Berkeley. Why don't you apply to Berkeley instead? And if you don't get into Berkeley, then you can go and implement plan A, which was to bum around Europe. And I got in, right? Miracles, I got in to Berkeley, and I did a PhD in plant biology. And I went there initially to work very much on plant development, on the genetics of plant development. But I ended up discovering how much fun it was to study not just the genetics of plant development, but also the evolution of plant development. And this was really the start of what has become my life's work, right? The thing that I, as at the beginning, it was just on a whim, like, oh, that sounds just as good as bumming around Europe. I might as well do that. And now it's become something that has become a really important part of my life and my personality. So I started at UC Berkeley. I did a PhD in plant biology, and I did my thesis on the evolution of flower development and the evolution of developmental genes. And then I moved from there to do a postdoc at Brigham Young University in Utah, because there was a really cool, although Brigham Young University is like the polar opposite of UC Berkeley in terms of everything about it is really different. And I never imagined myself being there, but in the end, there was a really cool job opening to study something super cool that was really closely related to what I'd done as a PhD student. So I just took the job and I went. And did a postdoc in evolution of development, and then started working in corn genetics, right? And that's what, on the left over here, you can see this is a flower of a corn plant, which is what I still study now. And then I got a job at UMass Amherst, where I'm now an associate professor, and where I've really continued this work. And my lab, we still study the genetics of development and the evolution of developmental genes, focused particularly on corn and other grasses. And so this is how I got here, right? And one of the questions that Matt asked me to answer was, how did I choose my field, right? And so I think it's pretty clear that it wasn't really that I had much thought in choosing my field. Instead, the field kind of chose me. There were two threads that ran through this from when I started as an undergrad at the university. It started as an undergrad at the University of Witwatersrand, and then just grew and grew and grew over time. And these two threads are one, that I think plant development is really cool. I think it's kind of amazing that you can go from a seed and there's something contained in that seed that can control the development of an entire plant. And the second thread was that I just find genetics and molecular biology fascinating, right? Although my number one love as a high school student was reading novels, when my dad said, no, you have to do a BSc, I wasn't completely bummed because I really did enjoy biology. And I also thought it was just really cool that there were these molecules that were regulating all of these things, like how I thought and how plants grow and how we get carbon, right? How carbon is fixed. And those two things were really initially just like these thoughts that like, oh, that's neat, I might as well do that. Oh, that's neat, I might as well do that. And that over time grew into something that I do now. I spend all my time thinking about these things, which in the past was just like a little like, oh, that's fun. The third reason why I ended up in this field, right, was, as I said, like my dad told me that I had to, that he wouldn't pay for university unless I did a BSc. And also, and I was pretty mad at him at the time, don't get me wrong. Like,
of course you were.
Yeah, right? But not so mad that I figured out how to pay for my degree myself, right? But and it was the third motivation was one of money, right? It's my dad's motivation was one of like, it's important to have a job that can pay the bills, right? My parents could afford to send me to university, but they definitely, neither one of my parents grew up very wealthy. They actually grew up pretty poor. And it was pretty important to my dad that we had good jobs that allowed us to pay the bills, basically, and to live a comfortable life rather than a hard life, right? Mm-hmm. And so this idea actually of my job as a professor, as a grad student, you're very badly paid, and as a postdoc, you're not very well paid. But my job right now is pretty well paid. I mean, we're never going to be as rich as this guy, but we have enough to live a very comfortable life. And I think that this is how did I choose my field? These things were the two important parts of it, right? Is I do something that I really love, that at the beginning, it was just like that's cool. And now is something that is super important to me and a super important part of who I am, right? And it wasn't back then, not by any stretch of the imagination, but it has become that, right? And so when I was in high school and when I was an undergrad, people used to say really annoying things to me, like, you should just follow your passion. And I was like, well, I tried that. My passion is reading novels. I was told I wasn't allowed to do that. And reading novels also just doesn't pay the bills. It's not a career, right? There are very few people in the world who've made a career reading novels. So that's dumb advice, right? Mm-hmm. To say follow a passion. But I'm going to say it now, right? You can follow a passion and get paid. And what I mean by that is that passion, when I was young, and when many people is young, it's not big enough to call a passion yet. It's just like a little thing that you think is cool. And there's lots of things out there that are cool, but there are some things that are cool and fun and exciting, and still get you paid, right? And it doesn't need to be one or the other. And that was the thing that I think was super important for me in my career. I felt like it had to be one or the other. Like either I had to be an accountant or I was going to be poor, right? And that was not really, I had to be bored and be an accountant, or I had to be poor and do what I loved. And you can do both, right? Which was something with hindsight that I wish I had known earlier on, right? Mm-hmm. The other thing that allowed me to do this, right, even though I was a bit of a flake and not the greatest student, as I said, I wasn't studious, but I did get good grades, right? And because I managed to achieve well enough that I could take each one of these steps, right, I could keep some of these doors open, right? And so I think that was the other part. I didn't put this as another bullet point on here, but I think this is the other part of my career that has been really important. is that I didn't know what I wanted to do, but my grades were good enough, and when I needed to get something done, I did it, and I did it pretty well, which meant I could keep these doors open that allowed me, as I was figuring out what I wanted to do and who I wanted to be, my options were open. So the next question I'm just going to go through this whole thing, and then people can ask me questions if they want to. The next question that Matt asked me to answer was, what does the future look like for plant scientists? So for me personally, I think the future for plant scientists in many ways looks really good, well, for botanists. And that's because of this, right? So this is a quote that I really love, and I always have in my my classes, and that's, when you look at where our carbon comes from, we North Americans look like corn chips with legs, right? And the reason why this is the case is that many North Americans eat meat, and our meat is corn-fed, right? Mm-hmm. So why would eating meat, right, that is fed by corn make us look like taco chips, right, with legs? And the reason why that is that every atom in your body, of carbon in your body, was fixed into a sugar molecule by a leaf, right? It's a thing that we don't think about all the time, but every single molecule of carbon in our body, right, came from carbon dioxide that was in the air that then was fixed into sugar by a leaf, right? So we basically aren't just leaves, right? We're the new version of leaves, right? And leaves do this. They make this sugar out of carbon dioxide through photosynthesis, right? So plants have this enzyme called rubisco and this process called photosynthesis that allows them to use the energy from light to take water and carbon dioxide and make sugars and oxygen, right? So this fundamental part of plant biology means that plant science will always be important, right? Understanding how plant works will always be a super important part of our world, right? Figuring out how to make more of agricultural products, of crops, figuring out how photosynthesis works so that we can start making it potentially synthetically so we are less reliant on plants. I don't think that will actually even ever really happen. But this centrality of plants to us, to the food that we eat and the air that we breathe, means that understanding how they work will always matter, right? So I think that this makes the future look pretty rosy in a lot of ways, right? But even if you don't care too much about plants, the skills that I teach are useful for anybody that wants to end up in biotech, right? And biotech industry is a huge employer in Massachusetts. And so I'm never going to not have a job, right? Especially now that I have tenure, right? I think there would need to be the collapse of higher education in the United States for me to lose my job at this point. Or if I did something really illegal, which I'm not planning on doing, so I'm not going to lose my job. But I'm going to be able to keep doing my job. And so that is good. Like, that offers some real career security, job security that I really like. And it's one of the reasons, apart from the fact that I saw that professors were actually paid pretty well in the United States, it was also the job security that was really attractive to me, right? In South Africa is very different to what it's like here. People don't move around in their careers as much in South Africa as they do in the United States. And so the idea of jumping between jobs and getting fired and then having to get another job, it was very stressful to me in a way that it isn't for many Americans. Not that getting fired or laid off is not stressful, but I think it's more stressful when it's not something that you were used to as a child, right? My dad worked for the same company from when he was, for his entire career. He got a job when he was 27 and he stayed working at the same company for his whole career. And that's what I was used to, right? And that was attractive to me, that I could go to one place and do a job and not have to change. But the buts, what does the future look like for scientists and for botanists? Right, if we acknowledge how important this is in our world and that the skills that we teach are useful and that education is important. The big but is that professor jobs are really hard to get, right? There are many people graduating, there are many more people graduating with PhDs than there are open positions for professors. And that's for lots of reasons. But two of the things that contribute to that is one, that government funding for science is low, right? We as a nation do not fund our science to high enough levels, right? It used to be really good, and now it's getting harder and harder and harder, right? To get public funding to fund the science. And most of the discoveries that have been made in the United States, part of the reason why the United States is so dominant in terms of its of science is because of this robust support for science that came from public funds. And that's getting worse and worse. And so that makes it hard to do the work. And it also makes it hard to justify employing more and more professors and scientists at university levels, right? The other thing that makes it hard is that there is a growing anti-science sentiment. This is something that I'm worried about. It seems like there's a growing anti-science sentiment in the United States and in many countries across the world, right? But those are big forces that are way outside of my realm and out of my control. And for right now, and for possibly the next 20 or 30 years, this is a really good career field to go into. And given these things, like government funding for science is low and there's a strong anti-science sentiment, I don't know what the alternative is there, right? Of like what decisions I would have made if I had known that that was going to be the case. I actually don't think I would have made different decisions because this job has allowed me, it pays the bills and it's really fun. And it's hard to think of other jobs that do that to the same extent, right? So that's how I got there. And this is what I see as being the future for plant scientists. And now I'm going to tell you one short little story about one thing that my lab studies. I can't tell you everything. There's too much to tell you. So I'm just going to tell you one short little story about genetics and flowers. So one of the things that we study is the development of unisexual flowers. So most flowers are not unisexual. They're hermaphroditic. Or bisexual. They have both stamens, which make pollen, which is the male part of the flower, and carpels, or the female part of the flower, which is where the eggs are produced, right? So pollen contains sperm, and the carpels have ovules that are the eggs, and the sperm and the egg fuse to give us seeds and fruit, right? So most flowers have both male and female parts in them. They produce both pollen and eggs. But in maize or corn, which we eat, right, corn produces separate male and female flowers. Most people don't know even that corn produces flowers at all, but it does. And the reason why we don't know that it produces flowers is because they're kind of tiny and boring. These are blown up to really high to show you what they look like. This is super high magnification. The flower on the left, that male flower on the bottom left, that's probably about 0.5 centimeter long, right? So these are quite small. And the female flower, there is an ovule down at the bottom, at the base of that flower that gets pollinated by sperm to give us the seed and fruit that we eat, right? So corn is actually fruit. So these unisexual flowers are super important for making controlled crosses of corn. So this is something that's happening happens all over the Midwest. And whenever corn breeders are trying to develop higher yielding kinds of corn, these controlled crosses happen. So what is happening here is that this scientist, this maize geneticist, has collected all the pollen in that bag, and then he just dumped it onto the female flowers that are in the ear, right? And he's going super slow here because he's showing you how you do this. And even super slow, you can see that it's really, really quick. So this ability to make controlled crosses for us as humans to decide where the pollen goes, right, means that genetics is really easy in corn, right? And the reason why that's important, why genetics is easy, why humans get to decide how these crosses are made, is that it means that we, and by we, I mean all of the maize breeders that have worked in the United States over the last 50 years, have been able to increase corn productivity substantially, right? So what this graph is showing you is that so on the x-axis, this is time. On the y-axis, this is average corn yield. And what you can see is that yield has climbed at this massive rate over the last 50 years. And this massive rate of yield increase was driven by the development of a single cross breeding strategy, right? Where that depended on the ability to make controlled crosses, right? So the reason why we have so much corn in the United States, so much corn that we feed it to animals, is because of this breeding strategy that was developed in the 50s and 60s that depends on having unisexual flowers, right? So the ability to make controlled crosses that's governed by floral sexuality has contributed significantly to yield increase in corn, right? So understanding how this works in corn, right? If we could understand how this works in corn, we could then use this knowledge to apply it to other crops that don't have these unisexual flowers, right? Which is other crops like rice and like wheat are also grasses that don't have unisexual flowers, which makes breeding rice and wheat much more difficult than it is in corn. So if we could figure out how to do this, we could start to control it and breeding would become easier, right? So we are studying the genetics of this. And I want to cut a long story short. Well, actually, I'm just going to tell you this anyway, right? That growth, this is what you're looking at here. So this is a male flower. So we study this, and I'm going to tell you a little bit about how we study this. So this is a male cornflower here that's at the top of the plant. Early on in development, the female parts of this male flower actually develop, right? The separate male and female flowers of corn that you see at adulthood, when they were baby flowers, they actually had all of the parts. But then what happens is that central, this is going to be, this is the carpel, this is the female part of the flower in this developing flower. Later on in development, that collapses, and it's just this little schmutz that's left at the bottom of the flower, right? And so these female parts of the male flowers form, but then they degrade. And so we get unisexual flowers, right? We want to know what are the genes that control this process, right? What are the genes that are turning growth off in these female parts of the flower that give us a unisexual flower, right? To find these genes, we use the reverse logic of genetics. And how this works, how genetics works, classical genetics works, is that when a gene is broken, right, like in this Drosophila mutant, this is one of the classical genetic mutants that was studied to understand animal development. When the wingless gene is broken, the flies don't have wings, right? Therefore, we know that the wingless gene controls wing development. So how we do this in Drosophila, in corn, in many different genetic model systems, we break the genes randomly using chemicals that mutate the DNA, right? So these chemicals are very dangerous because they mutate corn DNA. They also mutate our DNA. Our DNA looks no different to corn DNA to these chemicals, right? And they randomly, across the whole genome of corn, it'll break genes, right? And then we look at what happens after we've done that, what we call mutagenesis with this chemical, and we find mutants that are mutate, where the process that we're interested in is broken, right? So if you wanted to study wing development in Drosophila, this would be a great mutant to work with, because if you could figure out which gene had been broken, you would know which genes controlled wing development, right? So we did that with corn. And we made genetic mutants. We took a normal corn plant and we treated it with these chemical mutagens and found these mutants where the female parts of the flower are growing out of the male parts, right? So this is like that stuff that is at the top of corn, that like gross brown stuff, the silks. So now, instead of just coming out of the ear, these silks are coming out of the male part of the flower, right? because carpels are developing in the male flowers of these mutants. We call them Rapunzel because they have long, silky hair. And so because carpel growth is not suppressed in Rapunzel mutants, we can use the reverse logic of genetics to then say, therefore, this gene, Rapunzel3, controls carpel growth suppression. Right? We did a whole bunch of stuff that I can't talk about today to figure out which gene had been broken, right? Because that's the important thing to know, is that when you get a mutant out of this, you don't know which gene you've broken. You have to do some molecular biology to figure out which gene it was that you actually broke. And we did that. And we found that this gene, Rapunzel3, is an enzyme, encodes an enzyme that's really important in sugar signaling, right? And this is the first time that this has ever been shown, that sugar signaling plays a role in the development of unisexual maize flowers. Even though we've been studying corn and maize flowers for a really long time, this is the first time this connection between sugar signaling and growth has ever been found. And the questions that we're asking now in my lab is trying to figure out, is this specific to maize? Is this just a corn thing? Or is this something that is more general in the grass family? And that's it. I'm happy to answer any questions about this. It doesn't need to be about my work, anything about my journey, how I applied for grad school, what that process was like, what classes I took in undergrad, anything you want to know, I'm happy to tell you.
Go ahead, guys, and turn on your microphones if you want to ask a question.
Now.
Can you hear me okay? Madelaine, can you hear me?
I think you're still muted, Matt. Oh, no, sorry. I was muted. My computer was muted. Sorry.
I got it. Okay. Can you hear me?
Yes.
Perfect. Go ahead, guys, and unmute yourself there. I see you, Krish. One question I had is how, through the application of chemicals, is it not just like spraying a machine gun where you're knocking off a bunch of different, genes or whatever? How do you really specifically target stuff?
So that's an excellent question, and you don't specifically target, and you are hitting a bunch of genes, right? But the thing is that most of the time, so most of the corn genome, it's actually like tiny little islands of genes in a sea of what we used to call junk DNA, right? So the most of what you're going to hit with those chemical mutagens is not going to have any effect. To get such a very big phenotype, there are very few genes in the genome that will give you such a big phenotype. And then there's also things that we have to do. If you hit more than one gene that's giving you a phenotype, there are things that we can do to make sure to isolate that change from any of the other changes that happen.
Interesting.
Yeah.
Krish, did you have a question?
Yeah. So, are you a host for the program STEM at Work?
Yes. Usually I am, right, Matt?
Yes, you have been. Last 2 years at least.
Yes. Yeah.
Yeah. So I was just wondering what like a high schooler would do at your lab.
So because the high school is coming this summer, is it going to happen this year? Probably not, right? Or
there's still some debate about that. The state has withheld some of the funding to see if they want to release it at some point. It's not clear whether they will. But I think if I spoke with another professor and she said maybe there's creative ways that students could do some work at home and come into the lab under controlled circumstances to learn some skills. There may be a way forward.
There may be a way forward, yeah. And so in a normal year, absolutely. I'm going to talk about what a normal year looks like, right, to have students in the lab. We really enjoy having the high schoolers around. And so what you would do is, because it's the summer, that's when we are growing our corn. And so there's a lot of helping us with the genetics, crossing corn, making pollinations, like what I showed you there, helping us with DNA extractions, because we have to know, we grow hundreds of corn plants and we need to know what the genotype is. We need to follow these mutations through them. And so that involves a fair amount of molecular biology, of extracting DNA and doing PCR and gel electrophoresis. I don't know how much molecular biology you know, but basically it's like the basics of molecular biology to help us follow these mutations through genomes, and then the actual field work of helping with growing and crossing corn of genetics.
Wow, thank you.
You're welcome.
So you learn a lot of good lab skills, among other things. That's my impression from some of the final projects that students have submitted from working with you.
And you learn corn. You learn genetics. You learn genetics.
Yeah.
Yeah. Lab skills and genetics.
Yeah. I guess one question I have has to do with how adaptable is your field to responding to the demands of industry, for example? So if in the future, let's say this trend of government support for science is going to continue to drop, I know in some ways, especially in biotech, you have, big companies coming in with a lot of money and having the wherewithal to fund research. How adaptable is somebody in your position with a specialty in plant biology to be working on that, to make that kind of train switch?
Well, people so the same thing that's happening with biotech industry is happening with ag biotech, right? With the plant biotech industry. Mm-hmm. And people from my lab, I mean, right now I have the skills that you learn in my lab are directly portable to industry, right? Either ag biotech or normal biotech. I know a bunch of plant biologists, animal biotech. I know a bunch of plant biologists who are working in biotech in Cambridge, not necessarily in plant biology. So I was talking specifically about my own as a professor, but thinking about it, these booming biotech industry, right, in both agriculture and in drug manufacture and personal health, all of these things that are doing really well, and I think will continue to even under the pandemic, they need people that have the skills to be able to do this, right? Mm-hmm. Yeah. And so I think the job of being a professor and teaching people how to do this, I don't think is ever going to go away, right? And my staff getting direct industry funding, I do collaborate with some of these seed companies. It's the kind of thing that is possible to happen, right? But it is something that it's very different working in, I get to follow the questions that I want to follow, right? There's a lot of curiosity-based research that I really like and is actually really important for understanding the basics of how the world works, right? And so as soon as you go over to industry funding, that curiosity-driven research sort of goes away. And so I could do it, and I do it for some small projects, but I'd like to keep on doing this for as long as possible.
I think that's a valuable point to emphasize. Yeah. It's just you have a tremendous amount of autonomy, which is pretty rare to find in the job.
That's a good point. I forget about that, right? Because I haven't had a boss since 2004.
I mean, that's a blessing. I mean, I have, and I say that because I have a great boss. I've had a series of great bosses who just let me do my work, and it's just a wonderful feeling of freedom to be able to engage and to go in the direction of your curiosity on things.
Yes.
Karen, do you have a question by chance?
Me, I've been listening to the presentation. It's very interesting. I was thinking about a lot about what you said about like balancing passions with stuff that's actually going to pay the bills. I think, do you feel like science jobs, because this is something I've heard a lot, that like science jobs have a lot of how do I phrase this? Like communication and orally and written communication?
Yes. I think that was one of the things. So I was joking about how like what I really wanted to do was read novels. But one of the things that reading novels actually prepared me a lot for being a scientist, because you spend a lot of time writing and reading. I'm a very good reader. And I didn't realize at the time, while I was lying on my lilo in the pool, that like reading a lot makes you a good writer. And being able to read fast is something that is super, super helpful in science. And that's a skill that I developed just arbing around at home, lying around in gardens, right? It's a skill that I developed over my entire life, really. Yeah. And so that communication, both interpreting communication, oral and written communication, my reading comprehension is also very good. And I see a number of people whose reading comprehension is not so good. And I think that reading comprehension came from practice, right?
Yeah. I think, what I could add to that is the idea that we're probably happiest in our work when we play to our strengths. It's kind of the flip side of follow your passion. Well, your passion is also likely to be those things that come naturally to you, are easy, and which you have your curiosity just drives you forward more and more and more. And you're equipped somewhat naturally with all the necessary personal skills, which we call soft skills as I train these students, as well as, the hard skills of specific content knowledge. So I think I love how you told the story about how this profession sort of came and found you as a natural process of talking to people and other people saying like, you could go. I love the story about you could go to Europe, sure, but
Yeah.
how about try Berkeley, which is, that was a real stroke for you to have somebody step in like that. I think that's great.
Yeah, it was, right? And I was thinking that was another thing that as I was saying that, thank you for bringing that up again. It was something that was super important, was having good advisors, right?
Yeah.
The importance of having good teachers and good advisors, and that advisor was really taking, I didn't realize it at the time, right? I don't think I had shown tremendous promise in her lab, right? Like, I'd done well, I'd gotten A's the whole time, but I definitely wasn't her dream student, right? But she gave me the benefit of the doubt that, well, you got A's, you're kind of a flake now, but you got A's and you can write, so why not try this? And she just suggested this thing that was really life-changing, right?
It's incredible. And what I would encourage students to think about is, or just understand that part of what you're doing and going through education at all, whether it's high school, whether it's college, afterwards, even considering jobs, develop those relationships with people, because it is those relationships that are going to just open doors for you. Things are going to come your way. People are going to think, gosh, I have this thing I got to do, and so popped into my mind because I think they'd be perfect for it. That is how it works. It's just those sort of organic emerging connections that propel us forward.
The last thing I was going to say on that point, and it's also a very long answer to Kieran's question, was that my job now, and always has been, it's very much working with and talking to people all the time. When I was a high school student, I imagined that being a scientist was not a people job, that it wasn't something where I would need people's skills or need to be around people, and that's really not true. It's so much of science is team-based.
Yeah.
And it's collaborative, and it's like the oral communication skills and those soft skills, as you say, are super important, and you use them all the time.
I've got to say that's been really reassuring to hear, because as a kid I always heard about the lone solitary genius stereotype, but I one of my passions is creative writing. I do a lot of writing, and I was I really want to go into science, but I also want to be able to incorporate that. So it was great to find out that is in fact a big part of a lot of STEM jobs.
It's huge. I mean, yes, writing, being a writer and a reader is you're already ahead of most of the grad students starting their grad careers. To be a writer is, great.
It's a really necessary skill. Go ahead, Chris.
Yeah, so I've been curious about DNA replication before, and I know that polymerase chain reaction PCR that you mentioned is related to that. So could you talk a bit about that process?
What PCR is?
PCR, yeah.
So PCR is basically a way of photocopying bits of the genome, right? So PCR, genomes are really, really big, vast. Let me see if I can find this. I'm going to stop I'm going to stop presenting, and then I have another slide that illustrates this, that I think this is a thing that was really hard for me to understand why we needed PCR until I understood how big genomes are. So I'm going to find this thing. Bear with me for a second. Oh, there it is. So what PCR does, I'm not going to get into what the molecular biology of it is, but it basically makes thousands and thousands of copies, millions of copies of a tiny piece of DNA, right? Of 100 to 1,000 base pairs of DNA. And I'm going to present my screen so I can show you what the problem of size is. Can you see that? Yes.
Yep.
Right? So this is the area of the circle, and so here's corn, right? This is how big the corn genome is, right?
I think you need to, Madelaine, I'm not sure I can see it. Can you guys see it?
Yeah.
Oh, we
can? Yeah.
Weird. I don't know what to do. Let me try again. It says I'm presenting.
Yeah, and I'm not seeing it, so I just want to make sure it gets into the recording.
You might have another tab pinned, like on top of that.
Matt?
Yeah.
Well, I might.
Matt, because I can see it.
You can see it, okay. I don't see it. I only have one tab open. Wait a minute. Let's see. There we go. Got it.
You can see it?
Yep, got it.
Now I'm going to jump forward to where this slide was, right? So this is what PCR can do, right? PCR can amplify, and this was geared around sequencing, not PCR, which is why it says it's focused on sequencing. But PCR can make thousands of copies of a stretch of DNA that's about this size. Can you see my arrow?
Mm-hmm.
Of a long, it's about 1,000 base pairs, right? And the reason why this was super important, right, is that once you have thousands of copies of a piece of DNA, now you can start to manipulate it. This one piece of DNA, 1,000 base pairs of one piece of DNA, is really hard to work with. You need a lot of it to be able to see it, to be able to manipulate it, to put it into, to transform organisms, to do all sorts of things downstream. You need a lot of it, right? And for sequencing anything, right, the reason why we need to make thousands of copies of it is also to see it, and also so that we can focus on particular parts of these really, really giant genomes, right? So if we look at the difference between what PCR can do is 1,000 base pairs, right? The size of the corn genome, this is about the scale we're looking at. And it's hard to understand when we just look at the numbers. But if we look at what this means in terms of things that we're more used to, really big numbers that we're more used to seeing, right? So 1,000 square meters, that's like looking down on, this is from a video called Powers of 10 by Charles and Ray Eames. If we look at 1,000 square meters, this is what that looks like. As we get to 10 to the 8, right? This is the Earth that we're looking at, right? This difference in size, right, which is masked by this scientific notation where we just get 10 to the 3 versus 10 to the 9. It doesn't seem like that big of a jump. But each one of these powers of 10 in size is really making a really big difference, right? And so I'm going to stop sharing now. Because genomes are really, really big, right? Thousands, millions of base pairs of DNA in them. We need tools that allow us to handle that size, right? To either handle it in its entirety or break it up into smaller, more manageable chunks. And that's what PCR allowed us to do, right? Does that answer your question?
Yeah, thank you.
Yeah. It's just photocopying DNA.
When you say base pairs, is it 1,000 base pairs? So is that 1,000 of the exactly same base pair?
Yes.
Okay.
So, and that's what PCR will do, is you can choose a section of the genome that you want to copy a lot of it, and it's a series of molecular tricks that'll make thousands and thousands of copies of just that 1,000 base pairs. Does that make sense?
Cool. Yeah.
Yeah.
Yeah. Very cool. What do you think of CRISPR?
Well, I mean, we use it in our lab all the time, right? It's another tool like PCR that is a real game changer.
Mm-hmm.
It's suddenly we can do things like with PCR. When PCR was invented, suddenly we could do things that had before seemed like either had been extremely difficult or just outright impossible. Mm-hmm. And now CRISPR-Cas9 genome editing is the same thing, where it's like suddenly things are possible that I didn't think were going to be possible in my lifetime. Mm-hmm. And it's easy, it works, and yeah, we use it as a tool all the time. It's a very powerful tool. Mm-hmm. But like all tools, right? It's like a hammer isn't evil, right? But you might use the hammer to build a building that houses a really evil organization, you know? Right. It's like, it's a very powerful tool. You can do lots of stuff with it. It isn't inherently good or bad. It's something that allows us to do good things and also bad things, if that's how we're inclined.
Do you guys know what CRISPR is? Have you heard of it?
Mm-hmm.
You have, okay.
Yeah.
Yeah. I remember, I love tuning into science podcasts and, different kinds of shows where scientists get to really talk in depth about what they do. Yeah. And when I remember thinking when CRISPR came out, I was like, oh boy, that's a potentially threatening, very powerful technology. But it's not the first time we've opened Pandora's box.
Exactly, right? Yeah. And there are so many technologies that we've accepted, like our cell phones, right? Mm-hmm. I have this technology with me. I haven't thought about it at all, right? But it makes my life better, and it's a super powerful technology that I've just let into my life with almost no thought, right?
Yeah. Yeah.
And that's not good either, right? But it's like technology itself is not good or bad. It's what we do with it that is either good or bad, right?
Right.
Yeah. Yeah.
Do either of you guys have more questions for Dr. Bartlett? Dr. Bartlett, I want to thank you for today. This has been really great. It's good for me to see more in depth about what you do for your work, because you've been such a great site host for a couple of years for us. I really do hope we can continue this summer. It's just not clear yet.
Yeah.
But I'll definitely be in touch with you and let you know.
Yeah, great. And yes, I'm happy to take students. It's always a pleasure.
Yeah, awesome. Thank you so much for your time. I really appreciate it.
Thanks for coming.
Good thing.